español, entonces el seminario y la introducción va a ser en inglés, ¿no? Entonces sirve que practiquen su inglés. Okay, so welcome everyone to the seminar. Today we have Dr. Deniz Alacam. She's going to talk to us about modeling brain dynamics. I'm going to read a little bit about her experience. She got a bachelor's degree in applied mathematics at Ankara University in Turkey, where she's originally from. And then she got a master's degree in mathematics at Boston University. And then she got another master's degree, but in statistics at Georgia State University, and a PhD in applied mathematics at Georgia State University. Okay, she has 2 affiliations, well, actually more than 2. She is a faculty member at Bursa Uludağ University in Turkey, and she also has a research associate position at the Tri-Institutional for Translational Research in Neuroimaging and Data Science in Atlanta, and this is a center that has 3 universities, 3 of them. Georgia State, Georgia Tech, and what? Emory University. That's why it is called Tri-Institutional. Okay, and well, she has several publications, and her research interest lies in the use of mathematical and statistical tools to describe different brain phenomena. So we welcome you, and you can start.
Thank you for the introduction, and thank you for being here today. I and Rocio are we are friends from Boston University. We know each other almost for 15 years, and she's a great mathematician. I think you are really lucky to have her here, and thank you for inviting me here and letting me present my research here. So this talk was actually, there was a misunderstanding. I designed it for faculty, so if you don't understand things, don't get scared. So ask as many questions as you want, and because I will go in details of this subject tomorrow in the workshop. So as I said, if you have questions, if you want me to slow down, if you don't understand something, just let me know. Ask questions. I like questions. I like to answer questions, so don't be scared. Okay, deal?
Thanks.
Okay, so I will talk about modeling brain dynamics. My background is in mathematics, and I did a lot of modeling in using dynamical systems and bifurcation theory. Right now, I'm a research associate in translational research in neuroimaging and data science in Atlanta, Georgia. And my recent research is focused on fMRI studies, neuroimaging, and neurological disorders. What we usually do is we have like diseased brain and the healthy brain. We have different diseases like schizophrenia, Alzheimer's disease, or depression. So we look at what is the difference between the healthy brain and diseased brain. But this research is using mostly statistical tools, not actual math. So since I'm in the math department, I would like to present you some math work. So as you can guess, brain is really complicated. So it is hard to understand, especially human brain. There are billions of neurons and that much more connections. So instead of trying to analyze the complex brain, we start with something simpler. So here we will start with I will show you how to model smaller neuronal networks. Okay, thank you. So these small neuronal networks are called central pattern generators. They are, as you can see in the figures, they are made of really small number of neurons. It can be 3 neurons, 8 neurons, but it's not usually go over like 10 neurons. So it's usually like these small neuronal circuitries. So here, today, I will be talking about the bottom motif you see, 3-cell neuronal networks, and I will talk about how we model it. There is a complex way of modeling, and there's a simpler way of modeling it. So we will talk about the simpler way. And then how it generates rhythm, how it affects animal lives, we will talk about that. And if we have time, that's why I put it faded, if we have time, I will talk about a little more detailed model. And it's C-SLUG-SWIM CPG, I will talk about this. Okay? So what is a central pattern generator, or CPG? As I mentioned, these are small neuronal networks, and what makes them special is in closed circuitry, they can generate rhythms. So in the brain, when you poke an electrode into a neuron, what you get is a voltage value. I'm sure you have you ever seen EEG or EKG rhythms? They are just like ups and downs. There are peaks and they go down. So these type of voltage traces we get. So here, if these small circuitries are responsible for rhythmic behavior, like walking, breathing, swimming, or flying. In animals, they are responsible for this, but how they generate this rhythm, it's unknown. It's a mystery. So in this study, we are trying to understand how these neurons, how these circuitries are generating these rhythms. So here you see a C-slug. It's called tritonia. And what controls its behavior, swimming behavior, is this 3-cell network here. So these 3-cell networks are commonly seen in biology. So this is one example. Another example is this is called stomagastric ganglion. It's basically the digestive system of lobster. So you have seen a lobster, right? So in lobster's stomach, there are 3 teeth helping the digestion. One of them is on top, the other ones are in the bottom, the other 2. So how it works is the bottom 2 are in phase, they move at the same time, and the top one is anti-phase. So when the bottom 2 are active, it means that the top one will be inactive, and if the top one is active, the bottom 2 will be active. So this behavior is called pacemaking behaviors. Basically, the top teeth is controlling this behavior. There's a small video here. Let's see that. So this is a video from Al Silverstone lab from Emory University. So they work with lobsters. So here you see the lobster. So basically they open up the stomach of lobster, and now in a minute you're going to see, yeah, this is the stomach of lobster. As you can see, there are 3 teeth. One up here, and there are 2 on the sides. So 2 of them are moving in phase. They are in synchrony, and they are in the anti-phase with the top teeth, which is the 3rd teeth. So as I mentioned, this behavior is called pacemaker behavior, because the top teeth is triggering the bottom 2, so they are getting activated. And again, this behavior is also controlled by this 3-cell network. So that's why we are interested in this 3-cell network and how it generates the rhythm. So I gave you 2 examples, and there are multiple examples of these 3-cell networks. One of them was the lobster pyloric rhythm or stomagastric ganglion. The other one is tritonia, which it's a C-slug swims into like folding into 2 and then opens up. So there are 3 cells controlling the behavior. The other one is leech heartbeat model. So I will show you a video of this later on, but just keep in mind. So that's why we are interested in figuring out how these 3 cells are generating this behavior. So here is a snapshot of what we are going to do. This is a model generated by Hodgkin and Huxley model. Does anybody know what Hodgkin and Huxley model is? Do you guys know what a dynamical system is? Good. So, in a basic definition, so what does dynamic means? It means that it's moving, right? If a system is changing over time, we call it a dynamical system. That's the most basic definition of a dynamical system. So Hodgkin and Huxley type model, I did not show Hodgkin and Huxley equations here, but it's a four-dimensional model. The first variable is the voltage. So, and the second, third, and fourth variables are the gating variables. I will try to describe what they are in a minute. So, but since it's a four-dimensional system, it is, as you can guess, hard to run analysis on them. It's hard to analyze it, hard to look at the bifurcations. So instead of using Hodgkin and Huxley, in this study we are going to use Fitz-Nagumo model. Anybody heard of it? Yes, no?
Yes.
Yes? So Fitz-Nagumo is commonly used model. It's a two-dimensional model. It's simpler. As the dimension goes lower, it means that the system will get more simple and it's going to be easier to analyze. Okay? Any questions so far? As I said, don't be shy. Okay, let's continue then. So first of all, I would like to give you some background about the synapses, neurons, and their types. So I'm going to be talking about three-cell network here, but tomorrow, if you have time, I will talk about something even a little more complex. So for three-cell network, as I said, we use a two-dimensional network. Two-dimensional model, which is simpler, easier to analyze. And this type of models are called toy models. The reason they are called toy models, it gives us an idea about what's going to happen in the system, but it doesn't give us small details. But to model this network here, we will be using a highly detailed model, and I will show you tomorrow how to model this one. So we know that the brain cells are called neurons, right? And there are synapses in between them. So we can group these synapses in two different groups. One of them is chemical synapses, the other one is electrical synapses. So here what you see, inhibitory and excitatory synapses, they are called chemical synapses. The reason they are called chemical synapses, let's say that we have two neurons. One, we call it presynaptic, the second we call it postsynaptic neurons. And in chemical synapses, what happens is presynaptic, the first neuron, releases some neurotransmitters, which are, you can think of it as some kind of chemical or molecule. So what happens is, if they act like a key and lock relationship, so it goes to the next neuron, and if it is the right key, it unlocks a channel. And if the channel is unlocked, what happens is it opens up and the ions start to go in. So synapse get activated. So these ions can be either positive or negative, depending on its type, depending on the neurotransmitter. So it either supports the voltage in it or suppresses it. Okay? So if the ions are positive, we call it excitatory. And then in the diagrams we show it with this small triangle. If the ions are negative, we call it inhibitory, and we represent it with this small circle in here. And there is another type of synapse which is called electrical synapse. In electrical synapse, two neurons are actually physically attached to each other. There is a bridge in between them. And the ions are moving through this bridge. So, as you can see, since there is a physical bridge in between them, the synapses are really, really strong. And most of the time, 90% of the time, these neurons are highly in phase or highly synchronized. Okay? And another aspect of this neurons, they all have their own intrinsic dynamics. So a neuron can be quiescent. It means that it doesn't do anything. It just voltage level is stabilized at some level, and then it just stays there. It doesn't. You can think of it as it's sleeping. Or I think we are missing it. So it's 20 spiker.
There is a marker.
Okay. It is a tonic spiker. It it means that it's actively spiking. And each of these peaks are called a spike or an action potential. When I say a spike, please think of a peak just in the voltage level. So,
Yes, we have a question from
Sure.
a student from Adolfo, but he wants me to translate the
Okay,
question.
sure.
So the question is if there is some kind of relationship between the different kinds of synapses, if one of them affects the other one or vice versa. If you have a chemical synapse and an
Electrical at the same
electrical
time?
synapse,
It is possible, yes.
are they related? Is one affecting the other one?
Yeah, it can, but it is really rare to say like both like an electrical synapse and a chemical synapse at the same time, but they are affecting each other. So if the electrical synapse is strong, it means that sometimes it can make the chemical synapse maybe meaningless because it's weaker compared to that one. Because electrical synapse, the physical synapse, the two neurons are touching each other. Okay. But it's really rare. Any other questions? Okay. So this is what tonic spiker is. I don't know where it went, but and another option is the burster. So bursting is a mix of quiescent and tonic spiker. So it basically tonic spikes for a while and then goes quiescent, and then again tonic spikes for a while and goes quiescent. Okay? These are the type of synapses and type of cells we have. Any questions?
Can inhibitory synapse be burster type or quiescent?
Oh, anything. So this is the intrinsic structure. So this cell can be a quiescent cell, can be a tonic spiker, or can be a burster. And then it can still generate inhibition or excitation or electrical
So
synapse.
there would be like 9 types of different synapse behavior.
9 types.
Well, 3 for each. Well,
Oh.
inhibitory, excitatory, electrical, and
It depends on their intrinsic voltage level. Yes, you can, if you want to classify it, yeah, you can say that. But usually they're interested in just this type of synapse and the intrinsic behavior. So, yeah. So,
You want to put
no,
the last?
it's just I don't get confused. I think it's going to be fine. The figure came up. So, for each neuron, we use a model. It can be Hodgkin-Huxley model. It can be Fusio-Nagumo model. And also, there are ways of modeling the synapses. So here, if you look at here, you see this is what we call the fast threshold modulation. And this is how we define our synapses. So, are you familiar with these type of equations? Can you tell me what it's going to do? Any guesses? You can get some inspiration from the graph. So, if you look at the graph, you see that it's either on the top, in the maximum, or in the minimum. So, or you can just say that it's either 0 or 1. So, when you multiply something with this, depending on your voltage level, so if you look at here, for example, if whatever is inside here is, and your K is, just think of it as minus 100, minus 1,000, something like that. So if your K is big and if what is next to it is a positive number, it means that this is going to be a negative number and this is going to be big, right? It's going to go to 1. If it is, if the bottom part is larger, it means that it's going to go to 0. So it means that it's either 0 or 1. So which tells you that your channels, your ion channels are either open, or fully closed. So this is the easiest way of modeling a synapse. Do you think it is biologically relevant? Are the channels always completely on or off, open or closed? Hmm? No? So it is not biologically possible. It's not always, some channels are always on or off. They can be half open, they can be a little bit open, or they can be close to full open case. So, as I said, this is a toy model, and that's the reason we are using a simple equation here. But For a detailed model, we need to use synapses like alpha synapses or dynamic synapses, which is going to give us the flexibility to open it slowly and close it slowly or faster, whichever is the suitable for the model. And we have also something called pulse center oscillator. We have two neurons and they are inhibiting each other. So this is kind of closed, but so what we have is we have blue cell and green cell, and they are both tonic spikers. Once we turn on this inhibition, they become bursters. Maybe you can look at here, it's the same thing here. So burster, and they become anti-phase bursters. So basically they are like, when the green one is active, the blue one is sleeping. When the blue one is active, the green one is sleeping. So they're called anti-phase bursters. And once we cut off the synapses, they go back to the tonic spike region. As I said, this figure is blocking it, but you can compare it from this part. This is called half center oscillators. And why is it important? Because we have 3 half center oscillators in this 3-cell motif. So basically, we are going to be merging these 3 half center oscillators and create this motif. So here, as I mentioned, we are using Fitzhugh-Nagumo model. It's a 2-dimensional model. Our first variable is voltage. Second variable is the recovery variable. So I'm not going to go through these details that much, but I want you to take a look at the phase diagram. Do you know what a null line is? No? No? Okay, so a null line is a null line is this. When your variable is not changing. So remember that your variable is your equation is a derivative, right? So what does it mean if it is equal to 0? If your derivative is equal to 0, what does it mean? It's constant. It doesn't change, right? Because derivative basically means that it's changing over time. But if it is 0, it means that it's constant. So where it is constant is called a null line. So when you set this equation equal to 0, what you get is this S-shaped or Z-shaped curve. So this is going to give us one null line, voltage null line. For the other case, there is a second variable here, which is called the recovery variable. So this is going to give us a sigmoidal, this orange curve here. And these are called null lines, and the diagram that they form together is called a phase portrait. Okay? Do you know what a fixed point is? Yes? No? Can I see yeses? No? Okay. Fixed point is kind of similar to what I said with the null lines, but in this case, your variables both have to be 0. So just think of two curves, and they are both representing when the function or your variable is 0. So how you find the fixed point will be their intersection point, right? Because both of them are 0 at the intersection. Okay? And what happens here is we can basically follow our trajectory depending on our initial conditions. So these colorful dots are representing these cells in here. So we start them in certain initial points, initial conditions, and if the tail is longer, it means that they move faster. If the tail is shorter like here, it means that they are moving slower. Again, so green one and the red one are faster, and this one is slower. So why is it slow? It is slow because it's right at that knee of this null line. So, do you know what a bifurcation is? No? So, bifurcation means, let's say that you have a fixed point. And fixed points have their characters. They can be stable, or they can be unstable, or they can be half stable. So, what does stable mean? If your fixed point is stable, it means that whatever is around it, I'll show you an example soon, whatever is around it, whatever initial conditions you choose around it, they're going to move towards that fixed point. So that's why it's called stable, or it's called an attractor. if your fixed point is unstable, or if it is a repeller, it means that it's going to push away all the points around it. If it is half stable, some of them will go in, some of them will go out. So if you're talking about the bifurcation in a basic meaning, which means that our fixed points will change their characters. They can split into two, they can merge, they can become unstable while they are stable. So this is called a bifurcation. Okay? So, so here it is slow because, and we have a bifurcation parameter here, which is our I value. So what does I value do? I value basically moves this curve right and left. So imagine that this is going left. If it goes left, it means that there will be two more intersections, right? If there are two more intersections, it means that you're going to have two more fixed points. And if you move it slowly to the left, it means that these two fixed points will disappear. But there will still be a ghost of this fixed point here, and then that's why when the cells are passing through this region, they slow down. That's a rough explanation of what's happening. Okay? So this is the phase diagram of Fitzhugh-Nagumo, and this is the phase diagram of Hodgkin and Huxley model. As I said, Hodgkin and Huxley model is a complicated, compared to Fitzhugh-Nagumo, it's a complicated model. It's a four-dimensional model. So here you see a three-dimensional phase portrait of three-dimensional phase portrait of Hodgkin and Huxley model. So it's a video. Please keep watching. You see these dots. They are representing, again, they are color-coded. They are representing these dots. There's the red one here, and there is blue one and green one, which is stuck in that corner. So remember the lobster stomach. What the teeth on the top was a pacemaker, right? It was triggering the other two. It's the same logic. So here you can consider our red cell as the teeth coming from above, and the blue and red cells are the teeth coming from below. Okay, they are going to be in phase. So mathematically, what happens is this red cell is moving towards this manifold, and eventually it's going to go down, and then it will come up here, and it will push the null lines. It will separate them, so these two will escape. You can think of it as a let's think of it as a game, okay, guys? So let's keep it simple. So let's watch it. Let's see what's going to happen. You see the red one is moving. It's moving along the. Can you see what's happening here? Not really, right? So what happens is, as it's. Oh, I'm sorry. So as it moves here, basically it's generating a burst. So I can't see it either. You're supposed to see a burst here. And then it comes here, and then you see it basically separates these null lines, and then it releases blue and green ones. I think you can see blue and green ones. You see that they are in synchrony. So, and same thing happens now. Blue and green one goes there, and then it releases the red one. Then red one starts bursting. Okay? So this is basically what's happening. This is the application of theoretical equations that you have seen. And also, so is it the only rhythm that it can generate? No, it's not. So there are more rhythms that this these three cells can generate. So first of all, there are pacemakers. For each color, there is a pacemaker. There's also something called traveling wave, which they fire in order. But to analyze this, we use Poincaré return maps. Anyone heard of it? Okay, just think it's a theoretical tool. I'm not going to go in that much detail, but we're going to be using phase differences for this analysis. Phase differences, this is how we calculate it. We have a reference cell, which is the blue cell. We look at the burst initiation point, and then calculate the distance of burst initiation point to the second cell, and then we have this formula in here. We use this formula, and this is going to give us the phase difference between first cell and the second cell. Again, we do the same thing for first cell and the third cell. So this is how we calculate our phase differences. And as I said, that was Hutchinson model, and we do the same thing for Fitz-Nagumo model in here. And these maps are defined on the torus. You're familiar with torus? It's basically donuts or a bagel. You can think of it that way. So basically, there is a circulation on the torus. So the way we analyze this, we basically open this up. You open up and flatten your donut. Then here we have different initial conditions. And on the x-axis, you see the phase difference between the first cell and the second cell, which is the blue cell and the green cell. And on the y-axis, you see the phase difference between the first cell and the third cell, which is the blue cell and the red cell, I guess, right? Yeah. So once we start, I think this is, can we run this video? So once we start running this, it basically fills out our map. It comes from all the initial conditions and fills out our map. So now it's colorless, but now we're going to make it colorful to represent different rhythms. And here there are, I don't know if you're interested in computation, computers. So here we use GPUs, parallel programming. So when this was done for Hutchinson model, to get one single map would take around a week. Just this thing, just the thing you see in the board. But now we are using GPUs, which are parallel programming, so which gives us opportunity to run. For example, here I think there are 900 initial conditions. So it starts to calculate the equations for 900 initial points at the same time. So it gets just like a couple of seconds to get this map now. Okay. And this is again comparing Hutchinson and Fitz-Nagumo. As you can see, this is a complicated model, but and this is a simplified model, but they both give the same results. So as I mentioned, there are 5 rhythms which can be generated. So now let's make the map colorful. So each color you see here, the red, green, and blue, are representing the pacemakers. Just think of the lobster stomach. So here, in the first one, we have red pacemaker, so which means that the top teeth is the red one and the bottom teeth are the blue and green ones. And in the second one is green pacemaker. It means that the green is firing and blue and red are following them. And here we have the blue pacemaker, blue fires, and green and red are following them. Okay? Is it clear so far, guys? Okay. Then we have traveling waves. So we have 2 traveling waves. One of them goes clockwise, the other one goes counterclockwise. So the first one, the clockwise, goes like blue, red, blue, green, red, blue, green, red. It always like follows this rhythm. And the counterclockwise, which is the purple region here, the clockwise is the black region. It's the vice versa. So it goes like red, green, blue, red, green, blue. Okay? That's why it's called clockwise and counterclockwise. Are we good with these 5 rhythms? Any questions so far?
No.
No? Am I explaining very well? Okay, perfect. Okay, so what does this map mean? Let's talk about this. As I said, these are phase differences. And as you can see, they are varying between 0 and 1. What do you think, if I tell you that the phase difference is 0, what does it mean? What do you think? Phase difference is 0. Just think simple. It's nothing complicated. If the phase, remember what the phase difference, how we calculated it? We have a reference cell. We take the burst or spike initiation point and then measure it to the second cell, and then we had some kind of formula for that. So I'm telling you that that formula shows me 0. What does it mean?
It's there.
They are what? They are same thing?
They are in sync.
In sync, yeah, perfect, yeah. Of course, if the phase difference is, if the difference is 0, it means that they are in synchrony. They fire together. And what if I tell you that phase difference is 1/2 or 0.5?
Out of sync.
Yes, exactly. They're going to be out of sync. They're going to be anti-phase. One of them will be up, the other one will be sleeping, and then the other one will be up, the other one will be sleeping. Okay? This is how we read this map. So here we have an initial condition, and I think I forgot to mention in the previous one. These regions, there is a reason that they're color coded. For example, if you pick any initial point in this red region, it's going to merge to the red point, red fixed point, which is a stable fixed point. Remember what stable was? It was basically taking everything inside. And these regions are called basin of attraction. So they are all attracted to the red fixed point. And for here, they are all going to be attracted to the blue fixed point. And then the green fixed point, black, and purple fixed point. Okay? So here we pick an initial condition at the red basin of attraction. So which means that it's going to merge to the red fixed point here. So it's going to be a red pacemaker, correct? So if you look at it in the 2 dimensions, this is the phase difference between 1 and 2. We pick the fixed point in the, initial condition in here, and we see that it's merging to the top. Remember, this is a torus, so top and bottom is the same. It's all 0. So which tells us that 1 and 2 are in synchrony. They are in phase. And if you look at the second one, this is the difference between the first and the third cell. So basically what happens is it merges to 0.05, which tells us that they are in antiphase. Okay? So this is basically explaining what's happening. So if you look at here, this is our initial condition. If you look at the rhythm, it looks kind of like traveling wave, but not quite traveling wave. But so it starts from here and starts merging. So once it's merged to this point, this is the zoomed immersion, it creates a red pacemaker. Okay? And we can do this for other initial conditions. If I pick an initial condition in the blue region, it's going to give me a blue pacemaker. If I choose one in the green region, it's going to give me a green pacemaker. So this is another movie, basically explaining what I explained. Showing what I did in the previous slide. So we basically picked the initial condition in the red region, and you see that it's merging to the red fixed point because our red fixed point is stable. It's an attractor. So it'll generate a red pacemaker. As you can see, red is in antiphase with blue and green one. So here we choose another fixed point in the green basin of attraction, and it merges to the green fixed point in here. Okay? So which is going to give us a green pacemaker. Green will be in antiphase with blue and red. Okay? So again, this is another example. This is we picked another initial condition in the blue basin of attraction, and it merged to the blue fixed point, and it's going to give us a blue fixed pacemaker. Now, I think in the next one we will choose a fixed point from here. We'll see in a minute. Oh, sorry, it's down here. It's the counterclockwise phase, traveling wave. So it's gonna, as you see, we pick the fixed, initial condition from here, and it's merging to the fixed point in here. So it will give us an anti-clockwise traveling wave. And in the last one, we will choose a fixed point in here, and I want you to watch that one too. So this is basically how we use this tool. And there's a software for this. If you're using Linux or Mac, you can download it to your laptop. I think it should work. It's not working in my laptop right now, but maybe you can make it work, or I'll fix it soon, and then I'll upload the working version then. Again, which model do you think this is? Is it Hodgkin and Huxley or Fitzgerald-Dumont? Looks complicated. Look at these. Looks complicated or simple? Is it a single spike or a burster? Burster, right? So this is the complicated one. This is the Hodgkin and Huxley model. Okay, so this is basically how we use this maps. Now, I will mention briefly, don't get scared about bifurcation, okay? So, how we use these maps? What is a bifurcation? So, and let me show you one more thing here. So, here you see that there are extra points here. There are different points, and they are not getting merged, right? No trajectory is going in there. What do you think they are? I'm telling you that they are also fixed points. What can you tell me about their characters?
They're unstable.
They can be unstable, or they can be half stable or saddle, right? So there are these points in here. So as I mentioned, sometimes these fixed points change their characters. When they're stable, they become unstable, or there are two of them merged together and annihilate or destroy each other. So these type of behavior is called bifurcations. And we have two bifurcation parameters here. One of them is the external current in here. So basically, I can change it by hand. I don't need anything dynamic. I can plug in 0.1, 0.5, 1, 2, 5, whatever I want. So that's the bifurcation, first bifurcation parameter. And the second one is the Gij in here, which tells me the strength of the synapse. So some synapses are weak. I can take Gij as 0.001. Or some of them are strong. I can take it as 0.1. So in the x-axis in here, we vary the coupling term, coupling strength, and in the x-axis, y-axis in here, we vary the external current. So what happens? Let's see. Let's look at the first column. So the I here, our coupling strength is constant, and our external current is increasing. So let's look at here. How many different rhythms do you see in here? How many colors do you see? 3, right? And they are all the pacemakers, right? You don't see any traveling waves in here. So let's go up. When we go up, how many rhythms do you see in here? 5 of them. All 5 of them are here, right? We have green, red, blue, purple, and black. We have all of them in here. So if you look at this bifurcation diagram here, this tells us that, so this blue square represents pacemaker. So this dark green square represents mixed rhythm. It means pacemaker plus traveling wave. And let's keep going up. When we go up, what do you see here? How many rhythms are there? Just 2, right? Just the traveling waves, clockwise and counterclockwise. So if you, and do we have pacemakers? I think here on the side we have pacemakers. And if we continue going up, the pacemakers disappear and we just have white-green region. So this is what basically a bifurcation is. So you see that here there are multiple fixed points, so they can give, regenerate fixed points or they can destroy fixed points. So this is what a bifurcation is, okay? And we can do the same thing on the x-axis as we move along, as we move to the right. What you can see is here we just have pacemakers, here we have mixed rhythm, and here again we have pacemakers. So we do this for a large number of calculations. So we have here, it's a 75 by 75 grid. So you can think of like each of these dots in here is representing a map. Then we have 5,625 return maps and over 5 million traces. So it requires really strong computational power. Okay? So if you want to generate this with the old system, probably it would take maybe months. Because creating one of this was taken 1 week. But right now, I think we are able to create this bifurcation map in just 1 week. because, as I said, there are 5 million traces and then 5,625 maps. So it takes more or less a week or maybe a little less than a week. Okay? So what is next is we basically look at the bifurcation types. As I said, they can, when you talk about a bifurcation, they can annihilate or destroy each other, or they can create new fixed points. For example, here we have a stable. This is a bifurcation diagram, guys. I'll try to explain as much as I can. So the solid lines are representing the stable fixed points, and the dashed ones are representing the unstable fixed points. So what happens in a pitchfork bifurcation is all these three gets together and ends up with an unstable fixed point. So if you look at here, so can you see the gray fixed points beside the blue one? It's really tiny, but so the blue one is stable, as you know, it's the pacemaker, and the gray ones are unstable ones. So what happens is, as we go from this A3 to A4, as we increase the coupling strength, it undergoes a pitchfork bifurcation, and unstable saddles and the stable fixed point merges and creates just a single saddle fixed point. Okay? This is one example, and there are multiple ones. This is andronal Hoff bifurcation. So what happens is we have a stable fixed point, and as we decrease the external current here, we are passing from this dark green region to blue region. What happens is this fixed point undergoes an andronal Hoff bifurcation. While it was a stable fixed point here, it basically turns into an unstable fixed point, and it generates a limit cycle, which is both another dynamical systems term. You can think of a limit cycle as period, yeah, something like that. Okay. So let's skip this one. Let's skip this one too, and then let's talk about this. So we have different types of motifs. So, for example, if we say monobiased, which means that we are increasing just a single synapse, just a single connection, and then we analyze what's going to happen. Or it's pairwise biased, which means that we are increasing two reciprocal connections at the same time. Or we have this king of the mountain, what we call. So here, the outgoing connections from 3 is changing. We vary these. And in the clockwise one, we just increase the clockwise connections. So let's see what's going to happen. This is king of the mountain bifurcation, or king of the mountain case. So what do you think is going to happen? So the red one, there are outgoing connections from red one, and we are increasing these two connections. What's going to happen? It's actually in here, right? What happens is at the end, as we increase it enough, the red cell dominates the environment, so it all turns into red. If we can do it for green one, if the green is the king of the mountain, what's going to happen is it's going to all turn into green, right? It will be same for the blue. So this is how it dominates the system. Or if it is pairwise bifurcation, so there's something interesting happening here. We have what we call river. It's basically a limit cycle, or it's a trajectory going over the donut. It repeats itself. So why is it important? Why do we care? Because it helps us to switch between the rhythms. For example, let's say that we start from here, as if in here. This is the green pacemaker, right? Green will be in anti-phase with blue and red. So we start at green pacemaker, and as you follow this river, it goes to the blue pacemaker. Eventually, when we pick our initial condition around here, it becomes a blue pacemaker, as you can see here. Blue is in anti-phase with red and green, and then it goes back to it passes through the traveling wave region. Remember there was a traveling wave in here, and it becomes a traveling wave, and again it ends up in the green region, becomes a green pacemaker. So this is another movie. This is basically showing what's going to happen, what I explained in there. So it starts in the traveling wave region, it goes to the blue pacemaker, it passes it through, and then it goes to the counterclockwise traveling wave region, it generates that rhythm, and eventually it's go back to the green pacemaker, and then we will have a green pacemaker, and it's going to stop there. So this is called, so why is it important? Because we see this kind of behavior in biology. So this, what you see in the bottom, do you remember at the beginning I told you that the leech interheart neuron is also similar to three-cell network? So this is a leech. So as you can see, there's a blood circulation here. Now you're going to see that it's going from left to right in the bottom, from right to left. Okay? So it's going to do that for 3 times. And then after 3 times, you see that it changed the direction. It's exactly like in the mathematical model. It changed from counterclockwise traveling wave to clockwise traveling wave. So this is another, that's why it's important. Those rivers are important. So we have just 3 cells. What are they going to do? So is it enough to explain the brain dynamics? No, but what we can do is we can merge these 3 cells and generate larger networks. And we can understand what's going on in these larger networks. And these are other examples. So this is gastric milk. It's again a part of lobster. As you can see, there are 2 3-cell networks. And this is another example. And this is a pyloric rhythm. So you see that there are 2 3-cell groups. So once we understand what's going on in these small neuronal networks, we can put them together and understand larger networks. So that was the purpose of this study. Okay? Any questions? And let me finish. This is my group who helped to generate this research. He's my PhD advisor, Andrei Sholokhov. He's currently a professor in Georgia State University Math Department. And these are my lab members, my friends, my colleagues. I would like to thank them all. And thank you all for listening. And if you have any questions, I can take them.
Thank you very much, Deniz. Are there any questions?
Go ahead. Don't be shy, guys.
I can just try to translate if you want.
Well, I don't know. I think the students should ask first, but I'm just asking because I had a stupid editorial meeting and I came
Sure.
what exactly, I mean, the phase space in which you had your bifurcation, what did the axis represent there?
Those are...
Because I saw the notations, but I would not know what exactly they are.
In the bifurcation diagram, so it's a 2-dimensional bifurcation diagram.
Yeah, that's
So
obvious.
the base of this study is Fitsunagumo. Here. Fitsunagumo model. So in Fitsunagumo model, we have 2 bifurcation diagrams. One of them is in voltage, which is the external current. So you can think of, you have a neuron and then you are applying an external current with an electrode. So which is represented by the y-axis.
Okay.
And the second bifurcation variable is the connectivity strength. So here, for example, we are raising the connectivity between 1 and 2.
Okay.
So this is the second bifurcation
Got it.
parameter.
Thank you. Thank you very much. Yeah,
You're
because that's important to understand what we are doing.
Yeah.
More questions?
You can ask why is it blue, why is it red?
Yeah, did you think about the Daltonists who might...
Bueno, así, ya sabiendo todo esto, es posible replicarlo quizás, no sé, inicialmente. ¿O qué tan puede ser?
¿Compararlo con nosotros?
No sé, por ejemplo, todo lo de la sinapsis y todo esto, tratar de aplicarlo como por ejemplo a alguna inteligencia artificial o algo por el estilo, como para darles un poco más de realismo o más de...
Okay, so he's saying if is it possible to use this model and these rhythm generations and apply them to maybe artificial intelligence or neural networks in
Sure.
like computational neural networks?
Sure, actually there is a new study going on right now. So they have hybrid models, which means that some neurons are generated by these models and some neurons, the data is coming from actual animal. So it's trying to they do some kind of machine learning training in the system. So train their neurons as they to act like actual neurons. So it can be done. It's a work in progress.
More questions?
And in the workshop, my plan is, if everything goes well, so I will use an even simpler model. It's a one-dimensional model, theta model, and I have a MATLAB code. If you don't have experience in MATLAB, it's not a problem. I will give you the M file, so basically you will play with the parameters, and I will ask you to do things. For example, let me see. If you can generate King of the Mountain case, so how much do you have to increase? Sorry, how much do you need to increase these two synapses to get this? Things like that. Okay.
More questions?
And yeah, as I said, I will try to be much nicer tomorrow.
Okay. I have a question.
Sure.
Have you tried estimating parameters from the data?
Data? No, not for this case. We did it for other models, but not for this one. Because this is a toy model, so basically when we look at this analysis, when we look at this map, it gives us all the possible possibilities, all the possible cases, but we did not implement any actual data with this, real data with this.
But is it possible? Like
But, for actual data, usually these toy models, they don't usually work really well because they require more detail.
Okay. Okay, go ahead.
At the beginning of the presentation, you talked about you want to study like the brain disease, and I want to know if you already have like an example or a representation of any of those diseases.
I have some some graphs of the results of analysis, but it's going to be hard to pull out right now. But I can show them to you tomorrow or right after the talk. So what we usually do is we have fMRI study, so fMRI scans. They come from actual human beings in the United States. So we reshape the data into high-dimensional matrices, four-dimensional matrices, because there are thousands of subjects, and there are brain components, and each of them have different dimensions. So the matrices are big. Then they go through an independent component analysis. It's a higher level, maybe stats. Did you guys take statistics course? Do you know what the principal component analysis is? No? Okay, it's something to separate. So let's say that I have a signal. I have a group of data, and I want to separate them into independent groups. And these processes help me to separate them. So once they are separated, each of them define a component, and then, like in here, we are looking at the connectivity between these components. For example, like let's say that I have two components in here, and I want to look at the relationship between them. But instead of using differential equations, we are using statistical tools like correlation, functional network connectivity tools. And then what we do is we summarize them over groups for controls and diseased brains, then do a statistical test to see if the differences are significant or not. It can be a t-test, it can be some other tests.
you to go to Deniz's workshop that will be held tomorrow at 1 p.m. in room Aula Siglo XXI. Okay, you can go there. It's not necessary for you to fill a form. There was a form, and I think some people register in that, but if you haven't done so, no problem. You can still show up and attend the workshop. Okay? We would like to thank you again.
Just to finish, let me give you a little present from you.
Thank you so much.
This certificate, let me read it from you. Instituto Politécnico Nacional provide the following certificate to Deniz Alacam for the presentation Modeling Brain Dynamics as part of the first seminary of Applied Mathematics. Thank you very much.
Thank you all for your patience. I hope you learned something.